So my apologies for that. The good news is that there shouldn't be much on it. The really good news is that I think that by Tuesday we should be done with this stuff, which means we'll have Thursday to kind of have like a review class. So that's very exciting. Okay, so quick business. The exam average was 77. That's pretty phenomenal. That's really good. My goal is usually 75. So I was like, oh my god, it was probably too easy. But you know what? That's fine. That's good for you guys. A lot of people are emailing me to discuss the exam. Please come visit me. We can talk about all sorts of things. There's no recitation today. I am here. There's no recitation because I have no way to make up Tuesday's recitation. That's the reason for it. I am here until 5. So if you need to find me for whatever reason, you can. Next week is going to be an open recitation like we did before the exam last week. So my goal is to post practice problems this weekend, and then you have all next week to go through them, and then come to recitation if you would like to. Yes? Do we have a quiz this weekend? Yes, you have a quiz this weekend. Because we're still going to be covering stuff on Tuesday. So exam 2 is 10:28 to 10:30, so that should be on your radar. I hate when these things just kind of like jump up on me and I don't realize it's coming. So don't forget there's an option to take an oral exam. A number of people have talked to me about that, so now's the time to start really thinking about if you want to be one of those people. What I'm telling people is if you're the kind of person who's intimidated by the professor and is going to get like super nervous, don't do it, because that's, the brain doesn't work as well when you get super nervous. Or if you're the kind of person who's going to get, like, if you get shaken a little bit, then you're going to, like, blank out, don't do it. But if you're the kind of person who finds test anxiety of a written test so much worse, then that might be an option. Okay, I owe you a Stump the Teacher. My apologies. My goal is to get that exam graded and everything before I left. So I'll get that done today. Okay. Quick. Take 5 minutes to write. So the big way to study for this exam is to start organizing all this information into a kind of a compare and contrast kind of thing, starting at the top of the pathway with your receptors and your ligands, all the way down. And you'll notice things can be in different categories. 2 of the main categories we've been talking about are GPCRs and enzyme-linked receptors. And so I want you to start thinking this way. So I want you to spend some time right now to compare and contrasting signaling through these 2 types of receptors based on what we know, how they work. They're very, very different in the things that they activate and stuff like that. So go to it. Okay. Josh Carpenter. No. No Josh Carpenter today? Okay. Michael Iorio. I know who you guys are. There you are. Okay. You don't know? Is that what you just said? Okay, so I just want to know how you approach this. What did you think when you saw this question? Because this is actually going to help people a lot. Because I think half this class has the same answer as you right now. Okay, so that's great. So tell me, what did you think when you saw this? Were there any words that came to your mind, or any like, oh, I should probably be looking for this, but I'm not sure how, something like that? Or nothing, where you were just like, I'm just going to wait. Okay. Thank you for your bravery. Okay. Go ahead. Okay. So right off the bat, I'm hearing GPCRs, second messengers, enzyme links, either bind to a kinase or have intrinsic kinase activity. So yeah, so those are two very different things. That's exactly right. So, and that's because, okay, so second messenger. So what else? What else did he? I saw a hand go up over here. Colleen?
It would be like transfer.
Okay.
I also said that GPCRs, it releases GDP and binds to GTP. So an enzyme receptors don't use GTP.
Okay. So GPCRs require GTP and are regulated in that same way with GTP binding proteins. And enzyme link receptors are function independently of GTP generally. What kinds of enzyme activity do you tend to see with enzyme link receptors? Kinase activity, right. Okay, what else did people say? Anything else they were thinking of? Yeah.
I kind of made it like a table of like the receptor and where it binds, the process, and function, and started that with a.
Okay, so you made a table with receptor, what binds it, is that what I heard? And the process?
The process and like what result it has.
Okay, so the results from ligand binding?
Like the whole process.
Okay, so make a table. I think that's a good way to approach it. Make a table and start at the top and work your way down and think, okay, a ligand binds. And then this tends to happen with GPCRs, which would be conformational change, G protein activation, and activation of other enzymes, right? This is all general, right? This is not a specific pathway. With enzyme link receptor, you would say ligand binds, receptor cross-linking, so usually they tend to dimerize. And then you usually get kinase activity, either intrinsic or associated, which results in phosphorylation. And that creates docking sites for recruitment of other proteins. And so those already are two very different pathways, right? And I'm not giving you a specific signaling pathway. It's just kind of very general how these pathways work and how they differ. I guess the other thing that I should have put in here is nuclear receptors, because we did talk about that. So when you're studying for the exam and you go back and you look at this question, you might want to add in, just for your own study purposes, nuclear receptors. So you can, because that's also, if we have to put in nuclear receptors, we would say, how are they really different? Yeah. They're not on the cell surface. Right, they're cytoplasmic. Right, and their ligand is very different, right? Because, yep.
I was wondering, is there anything similar
Is there anything similar? Yeah, down. So like a lot of the activation stuff is very different. But as you work your way down the pathway, things tend to become more similar because really what ends up happening is a kinase somewhere gets activated, which somehow phosphorylates something that affects an effector protein. That's usually similar down at the bottom when you're talking about actually activating something. Yeah, all of them are actually. Yeah, so it's really at the top of the pathway where you have your big differences. So in nuclear receptors, right, you have a hydrophobic ligand that usually diffuses, or a small molecule that will diffuse through the plasma membrane and bind to the receptor. Some kind of conformational change that exposes the DNA binding site, right? So, kind of start studying. Okay, any questions about that? Thank you everyone who participated. Make a note of that. Okay, so far we've talked about all sorts of things. We talked about generic signaling pathways, and then we started kind of getting more specific. We pretty much started with receptors, nuclear, ion channel, GPCR enzyme. We didn't talk too much about ion channels, other than the fact that signaling through GPR sometimes does activate ion channel receptors. Is there confusion? I feel like I hear a hum of conversation. You know, it's so funny. So I was leaving my kid's school yesterday, and I was leaving, and I was talking with the woman who cleans the school, Miss Cook. She's awesome, and she's like, "You teach at Towson?" And I was like, "Yeah." She's like, "So is it easier to teach college kids because this is an elementary school?" And I'm like, "Well, yeah. I mean, I don't have to deal with any behavior issues." She was like, "Oh." And I thought to myself for a minute about how, like, my kids' classroom, when they go in the morning, it's kind of chaotic. There's, like, a lot of, like, kids who are, like, kind of bumbling around, trying to figure out what they're supposed to be doing, and kids pushing each other. We don't have to deal with any of that here. I'm guessing the kids who are the worst offenders might not have made it to college. That's possibly an issue. Actually, well, seriously, there's some craziness that goes on. But seriously, like, look at how you've grown up. You come in, you sit down. I don't have to tell you to be quiet, really. Or I do sometimes. Not really. So we had this, like, compare and contrast moment. Okay. Anywho, sorry, there was just a little conversation that made me think. I was channeling one of my children's first grade teachers. So we talked about mechanisms for turning off signaling. Specifically, we talked about GPCRs, how they work, how we turn them off, right, or arrestin and all that. We talked about different enzymes that are activated by GPCR, second messengers, example pathways, specifically the activation of adenyl cyclase and cyclic AMP production, how that works, and activation of phospholipase C beta, and release of calcium from the ER, and how that all works. And then we start talking about enzyme-coupled receptors and how they're activated, and that's kind of where we're picking up here. Right? So when you have ligand binding to enzyme-linked receptors, you get autophosphorylation of the cytoplasmic tails, and the goal of that really is to either activate other kinases and create docking sites for assembly of this whole signaling complex. So that phosphorylation, a lot of the time, is just causing some kind of conformational change or shape change that creates a binding site for recruitment of other proteins. And we kind of stopped on this the other day, right? We were talking about how we would inhibit signaling through this receptor. We talked about deletion of kinase domains, deletion of proteins, inhibition of signaling molecules. That's where we left off. So these intracellular signaling proteins that are recruited to phosphorylated receptors, they're binding to those phosphorylated receptors, specifically through interaction between the phosphotyrosine, because the tyrosines, well, in the case of tyrosine kinase associated receptors, the tyrosines get phosphorylated. And so you have a phosphotyrosine, a phosphorylated tyrosine, and that's the binding site for an SH2 domain in the other protein, in the signaling molecule. That's your protein-protein interaction site. So when I say phosphorylation of a receptor creates a docking site for it, what we're really talking about is the fact that it's creating that phosphotyrosine that the SH2 domain in the signaling protein is going to bind to. And that's either going to increase or decrease signaling depending on the protein that's recruited. If it's some kind of inhibitor, it's going to decrease signaling. If it's some kind of protein that's a kinase that's going to activate something else, or a GAF that's going to activate something, that might activate. It all depends. So really be careful about just being like phosphorylation, activation. No. So when I say increase or decrease signaling, what does that mean to you? Is that something that makes sense, or is you're just like increase signal? She says increase signal. Yes. So you're asking increasing, decreasing signaling, upregulation, downregulation, is that the same kind of thing? Well, an increase in signaling might result in upregulation of some process. I guess if that signaling pathway's job is to do that, I guess really when we talk about increasing signaling, when we're talking about increasing that transmission of the signal from the receptor to its effector protein. Upregulation, downregulation, often we're talking about gene expression, which sometimes those two are the same and sometimes not. Yeah?
This might be a stupid question.
No,
What
but
exactly is like a phosphotyrosine?
thank you. Who wanted to know what a phosphotyrosine was? See? These people are really happy that you asked. Okay, so what's a tyrosine? That's your question. Amino acid. Okay? So there are all these amino acids, and amino acids make protein, right? When you phosphorylate one of those amino acids, it's going to change the shape of the protein, right? You're getting a big electronegative phosphate group to the protein. Does that make sense? So you're like, well, what does that mean, right? You're saying to me, I see it in your eyes. You're saying, so what? What does that mean for anything? Well, the way everything works in biology, itself, in my version of cell biology, it's all about protein-protein interactions, right? So this needs to bind to this to make this thing happen, right? And so proteins bind to each other based on those biochemical characteristics of amino acids, right? Something that's positive is going to bind to something that's negative, right? So by throwing some big phosphate group on something, that's going to change that biochemical characteristic, right? And might either enhance binding, depending on what that other protein is. Maybe it has a spot that really likes binding to phosphate groups. And maybe it has a spot that doesn't like binding to phosphate groups. Depending on the protein, it's either going to encourage binding or inhibit binding. Okay? And so the SH2 domain is a specific binding site that loves binding to phosphate groups stuck onto tyrosines.
Yeah, I think in the physiology class we learned that like tyrosine can become certain hormones based on what enzyme it like binds to. Is that probably
Tyrosine can become a hormone based on what enzyme it binds to. Oh God, somebody's got to teach that to me. My physiology is like, whoa. But you might be able to teach me. Yeah.
So what did you say about the increase in signal?
So what we're talking about is, we're talking about recruitment of all these proteins to the phosphorylated receptor, right? Sometimes it will increase signaling, meaning it will help in relaying the signal down the line to affect the effector protein. That effector protein could be a transcription factor. In the example I tend to give the most, so it may activate gene expression. Decreased signaling will be the opposite. Okay, any other questions? Do people feel a little bit better about understanding phosphotyrosines and SH2 domains? Please see me if you're not. It's one of those humps I think people have to get over in cell biology, is it's getting to that point of intuitively understanding protein-protein interactions and how they affect function. That's actually something that you don't really intuitively think about until like it just clicks one day. So if it's not clicking for you, you should see me awake and talk, or ask your neighbor. Yes.
These are with like the enzyme link receptors.
These are yes, we're talking about enzyme link receptors. And the reason you know that is because when you're looking at this receptor, is that a GPCR or an enzyme link receptor? Right, because a GPCR you would see a 7 transmembrane pass associated with a G protein. Yeah. Yes?
Is this applicable to all enzymes, or are we still just talking about the RTK?
So here, okay, so the question is this applicable to all enzyme linked or just RTK? It's all enzyme linked receptors tend to get phosphorylated and there are docking sites and recruit other signaling proteins. RTK, it just so happens that we're talking about intrinsic enzyme activity and phosphotyrosine. Sometimes you'll have other kinases but still phosphorylating. Sometimes you'll have a serine kinase that will phosphorylate on a serine and that will recruit a different. It won't be an SH2 domain, it'll be a different domain that binds to that. But the general idea of phosphorylation and recruitment of signaling proteins that's true for all. Okay. How are we doing? Is everyone happy? You know, it was really nice about going to Florida. I forgot about that nice Florida breeze. You know, you like get there and it's like hot and sticky, but then you like smell the ocean. And you're like, oh. I grew up in Florida. I was in there forever. Anywho, okay. I've been dreaming about the Florida breeze apparently. Okay, so let's talk about another GTP binding protein. RAS. I told you it was coming. So RAS, what it does is it relays a signal from the cell surface to different signaling pathways. And so you can make all the assumptions that you want about RAS based on the fact that I'm telling you it's a GTP binding protein. That means that when it's GTP bound, it's active and it probably can bind to something and do something that's activating. When it's GDP bound, it's inactive, it's off, it cannot do anything, it's not relaying the signal. You can probably guess how the activity of RAS is regulated. How do you think that activity of RAS is regulated? Gaps, right? What do you think the effect might be if RAS is mutated in such a way that it cannot bind to its gap? It'll be on all the time. What do you think that effect will be on signaling? Will signaling be on or off? Will be stuck on. Is it surprising that a RAS is pretty often mutated in cancer? Not surprising, right? Because lots of signaling, dysregulated signaling is associated with cancer. So it could be at the level of the receptor. The receptors are dysregulated and always on. It could be at the level of signaling proteins, where proteins that tend to relay the signal and need to be on or off or stuck on. It could be at the level of phosphatases that might turn things off by removing phosphate groups. Really? We're just screwed. It's kind of amazing we don't all have cancer right now. Huh? 1 in 3 people will get cancer. Is that 0 here? It's possible. I believe we're always on the verge, and we just have really good ways of doing it. Yeah. Right, because what does GAP do? Okay, good. Sometimes people confuse GAP with GTP. Good. Okay, questions about just RAS and what it is? We're going to talk a little more. Yes? Is RAS in all cells or in specific cells? I believe RAS is in all cells. It's just whether or not it's stuck on is specific to cancer cells. All right. Other questions? I saw another hand going up. Okay. So RAS's activity, just like we spoke about, is activated through recruitment of a RAS gap to the RTK signaling complex. So actually how it works is RAS is hanging out associated with the plasma membrane. It's there on that inner side of the plasma membrane in close proximity to all these receptors. It's always there. But the reason it's not always signaling is because it's usually in the GDE bound state. And when you get all that phosphorylation that occurs when a signaling molecule binds, one of those docking sites that's created is a docking site for this adapter protein, which recruits RAS-GAP. So you're getting recruitment of your GAP, so you can activate RAS. So that's how that works. And so then when RAS is on, what it does is it kind of spreads the signal to multiple pathways. So RAS doesn't just, don't think of it as just this linear activation. It actually activates in multiple directions, and which is why it's super bad, super duper bad if it's mutated. It's not good. So when you're looking at this kind of figure, you want to ask yourself, how is RAS's activity regulated? Through GAPs and GAPs, right? Specifically through recruitment of the GAP to the phosphorylated receptor. If there's no signaling molecule, you don't have this phosphorylation, then you don't get recruitment of your GAP, so then you don't have activation of RAS. Signaling is off. Why isn't RAS active all the time? Well, normally when there's no signaling molecule, there's no recruitment of the GAP, right?
Is RAS activity exclusive to GPCRs? Sorry.
Is RAS's activity exclusive to enzyme-like receptors? I believe not. I believe I think there's some crossover. I mostly think of it as associated with enzyme-like receptors, but I feel like in my mind I'm seeing an example where it is not, where it's also involved. Give me a minute. I feel like I might have a slide. Any questions about RAS and why it's only on in certain cases, what phosphorylation has to do with it, how RAS is activated, how signaling molecules binding to receptors activate RAS? Yes?
So what would the mutations look like?
So tell me, what do you think? What kind of mutation would keep signaling on all the time? You can come up with a few probably. And you have to choose which protein you want to mutate. So let's just kind of, what you want to do is look through each protein in this kind of figure and say to yourself, well, what might keep signaling always on? If we look at, the most obvious two to look at would be RAS itself and the GAP, right? Or even something with the receptor, right? If the receptor always was phosphate, let's say the phosphatase that would remove the phosphate group was inactivated somehow, which allows group 2 to always be attached and always be interacting with RAS-GAP, then you would always be on. Or let's not forget there's not just turning on, but just turning off. So you don't see in this picture, but there is a gap that's going to turn it off. So let's say RAS is mutated in such a way that the gap wouldn't work, so it'd be stuck down to GTP, then it would always be on. So those are 2, 3 examples. Yes?
The activation of RAS so I can see if I get it.
Yeah.
So the signaling molecule binds to phosphorylated receptors, which is going to include the adaptive.
Wait a minute. I'm stopping you there. You said the signaling molecule binds to a phosphorylated receptor. What's wrong about that? What happens first, signaling molecule or phosphorylation of the receptor? The signaling molecule turns on phosphorylation of the receptor. So that's very important there. Because without phosphorylation of the receptors, it makes the docking site. So if you have phosphorylation of the receptor, then you're going to have signaling on regardless of your signaling molecule, and then you've lost your regulation. Receptors are, their activity is regulated, or signaling pathways are regulated often by ligand binding, which kind of then the whole thing goes down the line, turning it all on. Something's funny out there. Yes?
I'm not sure if this is or not, but is there something that causes the signal molecule to leave the receptor that would then make RAS turn off?
So your question is, how does this get turned off?
Yeah.
Does a signaling molecule leave? So remember last week, or maybe the week before, we had a figure which showed all the different ways to turn off signaling. And then we did one in recitation with I guess. That wasn't turning off, that was actually turning on. But still, remember we learned about receptor-mediated endocytosis.
Okay.
So you can endocytose your receptor to turn it off.
Okay.
You could also have phosphatases that are going to remove your phosphate group. That can turn things off. You can have inhibitory proteins that are recruited that can turn things off. As far as signaling molecules not binding anymore, I think more often than not, once they're bound, but I believe what happens is they're endocytosed. Yeah. Okay, go back to what you were doing.
So signaling binds to the receptor. Receptor is phosphorylated, creating the docking site for the adaptive protein. And then the adaptive protein recruits the GF. And then that's where I'm stuck. I don't know how the GDP gets released.
The gap. The gap
So
does.
the GF is gonna bind to GTP.
Probably what it's doing, it doesn't show it very clearly here. Probably what it's doing is interacting with RAS, causing some sort of conformational change that releases the GDP and allows GTP to bind. So what Tammy was just saying here, and you guys are always welcome to come to my office and go through it all, or raise your hand in class and walk through it to make sure you understand something. So she said, okay, signaling molecule binds to the receptor, it gets phosphorylated, creates a docking site for an adaptor protein, which recruits the GEF. Then she got confused as to how that is associated with RAS turning on. And so remember what we've learned, and pretty much how all GEFs work is they're going to somehow interact with the GTP binding protein, cause a conformational change that releases GDP and allows GTP to bind. That's not very clearly depicted in this figure. Yes?
I always kind of thought GDP turns into GDP.
No. Yeah. So a lot of people, I saw that actually on the exam, a lot of people thought GAPs phosphorylated GDP. GAPs are not kinases. They're not kinases. They actually just release the GDP and allow GTP to bind. They are guanine nucleotide exchange factor. They're exchanging.
So is GAP releasing GTP?
GAP, no. So GAP is actually an enzyme that's hydrolyzing the bond that's removing that phosphate. So it's actually cutting that last phosphate group off. Yeah. Okay. How's it going? Do we love RAS? Yes.
So GTP turns into GDP through hydrolysis, but GDP gets just exchanged with GTP.
Yes,
So it doesn't
GTP turns into GDP through hydrolysis of that one phosphate group, release of that phosphate group, yes. But GDP does not turn into GTP, it's just released and GDP is present in the environment it's environment. Yes, at some point in time, somewhere, other enzymes make more GTP. It comes from somewhere, but it's not happening right here. Yes.
So would you think of it as like when you do a conformational change, another domain is showing for?
Yes, so that is the way to think about it. When you do a conformation, so the protein makes a conformational change, which what it does is it makes the binding to GDP not strong. And so it's released. And it makes the binding for GTP stronger. So it can bind. It's as simple as that. Okay. So I keep talking about cancer. Any other questions before I move on? So I keep talking about cancer. I love talking about cancer. It's so much fun. And so just this is the RAS signaling pathway. And so you notice a lot of, I just wanted you to have a feel for how busy it is. There are a lot of things that are activated. Remember, because this is a protein that spreads a signal along multiple pathways, activates multiple pathways. So outcomes of RAS signaling can be proliferation, so cell growth, cell survival, differentiation, effects on the cell cycle, again, cell growth, cell motility, tumorigenesis. Well, tumorigenesis is kind of like the ultimate outcome there. And so anytime you have too much cell growth, too much cell survival, too much cell cycle, all these things, cell motility. Motility is really big for metastases, right? All of this is going to be really favorable for cancer formation. And so like what we were talking about before, often these tumor-associated RAS genes are always on because they have point mutations in the RAS gene. Okay, so you've got RAS is on, it's going to spread its signal through multiple pathways. And so usually what it does is RAS is going to activate a MAP kinase. And here it gets kind of like, who cares, because there's MAP kinase, MAP kinase. It all sounds the same, but basically just think of it as these MAP kinases that phosphorylate each other and activate each other. And there's 3 of them, right? 1, 2, 3. And they kind of work together. They do work together. And that they're going to affect some effector protein. They're kinases, so they're always phosphorylating something. And so they're going to phosphorylate these different proteins, and it might be a protein that's going to change the activity of that protein. It might be a protein where that's a gene regulatory protein, so it'll go activate gene expression. But remember, phosphorylation is always pretty much affecting protein activity in some way, in either allowing it to bind to another protein, or allowing it to go into the nucleus, or allowing it to go into the nucleus and bind to some other protein, or phosphorylate some other protein. But it is relaying that signal, is what it's doing, through that phosphorylation. Okay, so let's do practice problems. So I feel like you guys need a little bit of change of scenery. So you're studying pancreatic cancer. So since RAS, we just talked about, is frequently mutated in cancer, you decide to sequence the RAS gene in your cancer cells that you're working with. Right, so you got cells from your boss who's like, we don't know what's wrong. So you decide, okay, well RAS is always mutated. Let's sequence the RAS gene, see how it looks in these cells. And check for different mutations. And you find a common mutation in your tumor samples that results in a G2B mutation at position 12. Position 12 meaning the 12th amino acid. G2B, glycine to valine. A quick look at the literature, right? Because what do you know about that? Nothing. So you go to PubMed and you do some reading. A quick look at their literature tells you that this mutation falls into a gap binding domain. Hypothesize the effect of this mutation on the RAS protein function and the effect on the signaling pathway. So how does this affect RAS itself? How does this affect the signaling? Is this an activating mutation? Is this an inhibitory mutation? Is this mutation possibly contributing to the development of cancer? And then design an experiment to test your hypothesis. Okay, so take a minute to talk to your friends about this. I will circulate, clarify if you need it. So here. Okay, so Okay. I see that people are just packing up and leaving. which means that they're bored. So I'll try not to spend so much time on practice problems. I like to do it because I know it gives opportunity for people to really talk about things and stop listening to me talking. And also call me over if you have a question, because not everyone feels comfortable raising your hand in class. But then I just saw a whole bunch of people pack up and walk out, and I was like, okay. Maybe it's because, and they'll hear that hopefully when they listen to this and go, oh my god, I should have done that. It might be because there's no recitation, so people knew that and have their job there. But anyway, so what was the answer here? I'm not going to call on people because half the class is not here. Yes. Okay, so yeah, so a number of people listened to the lecture and where we did this before, and yeah, mutation back. So let's go one by one. Okay. So you see that the mutation falls into a gap binding domain. So that means the gap cannot bind. You think the gap probably cannot bind to RAS. This is called forming a hypothesis, right? And so you think to yourself, well, if the gap cannot bind to RAS, how will this affect the signaling pathway? Will it be on or off? On, right? Because it'll get stuck on. You won't be able to hydrolyze the GTP and turn it off, right? And is this mutation contributing to development of cancer? Possibly, because you have signaling on all the time, then shut off, it's likely to result in cancer. And so the hard part is designing an experiment, right? And so what I heard just up here is mutate the mutation back. Well, really what you would be doing is changing that point mutation back to what's the wild type version. You know this, the human genome sequence, you just look it up, you say, okay, right, this was supposed to be a glycine, I'm going to change it back to a glycine, and see what happens. Yes? Right? So you would do both ways. You would take the cancer cells and you would overexpress RAS that has the normal sequence, or you would take normal cells and you would introduce the mutant and see what would happen. Right? And so what would be your readout? What would you be looking for? I heard something in the back about looking for expression of a gene or a protein that you would expect to be made as a result of activation of this pathway. So with the wild type version of RAS, it should get turned off, right? So you should have lower levels of that protein. Yes? So, okay, so that's exactly. So you remember I was saying exactly what RAS does, and that's exactly what you do. You go back and you say, what does RAS normally do? What would I predict it would do if it was out of control and always on? What would I predict it would do if it was normal? And one was cell cycle. So you would look at cell cycle. And actually, at the end of the semester, I think the last unit we do is on cell cycle, and we are going to look at ways that you can look at cell cycle, like actually experimentally ask that question and evaluate that. So one was looking at expression, one's looking at cell cycle. Did anyone have any other thoughts? Huh? Cell growth, yep. Cell cycle, cell growth, both are related. Yep. Yep.
Well, just a question, is it activating because it activates?
It's activating because it's keeping the pathway on. Yep. Inhibitory would mean it would keep something off. Yeah. Any other thoughts, questions? If you had a hard time even thinking about how to approach this, if you did, feel free to either ask any of your friends in class who you feel like maybe are having an easier time understanding this, or come see me and we can talk through it. I can tell that people are getting better and better at thinking in this kind of scientific way. The real challenge is knowing the question that's being asked and making sure the experiment that you're thinking of is actually answering the question. Yes?
If it was the other way around, would it still be considered cancer or a disease?
If it was the other way around.
Like if I had a mutation in GDF instead of GAP, so it would become inhibitor. So would that still
The question is, if there was a mutation that inhibited deaf activity, would that cause cancer?
I'm not sure.
Because then you would say it wouldn't turn on.
Yeah.
And so you think, would that just cause some kind of disease? Maybe, or would that individual even be viable? Would that individual ever exist without a functional RAS? Probably not, because RAS is so important in relaying so many signals that you can imagine that if you've got a mutation that prevents RAS from ever turning on, I'm guessing that you'd never be born. But I don't know. It's a hypothesis. You know how you would test that? You would make a transgenic mouse. See if you ever get a mouse, or if it's embryonic in there. Any other thoughts? Okay. I think we'll stop here just because I want to have, we'll have to have plenty of time to get through the rest of this. Okay.